Good day. Today, we will be learning about the microscope. My name is Ray Valencia. Here are the objectives of this lesson. To identify the different parts of microscope and to determine and explain its parts. Let's find out what are the vocabulary from this lesson. Let's start with microscope, revolving nosepiece, objective lens, clip, diaphragm, light source, eyepiece, arm, slide stage, horse focus knob, fine focus knob, and base. Let me ask this question. In your own opinion, why some people are wearing glasses? What do you think? All right. That's good. Thank you. As we have learned that organisms are made of billions of cells. However, most cells are too small to be seen by our naked eyes. To see cells, we need a microscope. And this is what are we going to learn today about the different parts of the microscope and how these parts function to see our cells. Before we proceed with our microscope, let me just show you this onion microscope slides.
Preparing onion microscope slides. You will need the following materials: a microscope camera, microscope slides, a kitchen knife, toothpicks, pipettes, cover slips, iodine tincture, an onion, and a cutting board. Preparing the slides. First, wash and peel the onion. Then, slice the onion as thin as possible. I found that using the thin layer of skin around the onion worked best. Clean your microscope slide with an alcohol wipe. Do the same with the cover slip. Place your pieces of onion onto the slide and reposition them with a toothpick. Add a drop of iodine onto the onion. Drop on the cover slip. Apply pressure to the cover slip, but not enough to break it. Now your slide is complete. Now observe under a microscope. You will be able to see the cell wall and the nuclei of the onion cells. The onion's rigid cell walls allow the cells to be viewed under a microscope, and the iodine dyes the nucleus red, allowing it to be seen.
All right. You have just seen the video about getting the or showing or looking for the cells of the onion. What is the purpose of the experiment? These methods allows the student or you who are watching right now to view plant cells under the microscope. We know the purpose and function of microscope, right? A single layer of onion cells membrane can be easily obtained from the bulb. This can be stained to bring some cells structures, right? We have the cell wall, cytoplasm, including the nucleus and cell membrane or wall, into contrast so that they can be observed using a light microscope. All right. So, and also, okay, why they used onion? Of course, there are so many plants that we can use, but why onion is the best way or the best material to use? Because onions are multicellular plant organism, which basically means that they are made of many cells. When the plant or our organism is made up of many cells, it is called multicellular, that are uniform in size and shape. Not only multicellular, remember that, but uniform in size and shape. Because unlike animal cells, which appear a lot of more differentiated, even though animal cells are multicellular organisms, but because of the uniformity, when it comes to uniformity, they are not the best way example for this microscope experiment. The uniformity of the onion cells are due to the existence of the cellulose, which helps the cell maintain its shape. Remember this, animal cells appear a lot of more differentiated cells, unlike an onion has uniform in size, which are uniform in size and shape. All right, now let's find out the animal cells using chick cells from human through microscope. All right. Remember this, guys, we are using the iodine solution because a small square or circle of thin glass called a cover slip is placed over the specimen. It prevents the slide from drying out when it's being examined. Iodine stain can be used to stain plant cells or animal cells to make the internal structures more visible. Remember this, we use this to make the internal structures of the cell more visible. All right. Okay, these what we can see now are the cells of these chicks, human cells from coming from the chick of a human. So we have the nucleus, the most important part of the cell. All right. You may ask yourself why we use the chick cells for the observation in microscope. Because the purpose of this is chick cells are eukaryotic cells. The eukaryotic cells are, the cells that contain a nucleus, okay, and other organelles within enclosed in a membrane that are easily shed from the mouth lining. Therefore, it is easy to obtain them for observation. All right, now this is the anatomy of a microscope. We will be able to find out and understand the function of each part. All right. Now, this is the body tube. All right. The body tube connects the eyepiece to the objective lens. Eyepiece, okay, then connect to the objective lens, this one. All right. Now, arm connects the body tube to the base of the microscope. This is the arm connect to the base, and this is the power. Okay, arm connect to the base of the microscope. Next, eyepiece over here. So the eyepiece is the lens that viewer looks through to see the specimen. Okay, the eyepiece usually contains by 10 times or 15 times power lens. All right, eyepiece. And now, coarse focus knob brings the specimen into general focus. So in this bigger one, cylindrical part, so this is the coarse focus knob. It brings the specimen into what we call this one, into a general focus. All right. Now, how about the fine focus? Fine tunes the focus and increases the detail of the specimen. So with this, so you are adjusting into a closer part of the specimen. Okay, what we had earlier, part of onion cells. So if we are going to use this microscope, so using this one, this coarse focus knob, this will be a general, okay, look of focus. But using this fine focus, it will take you closer to the specimen, which is the onion. All right. Now, revolving nose piece. All right. Rotating turret that houses the objective lenses. Okay, we have three. Some microscope has two lenses. Some has one, but now this is the latest one. They have three lenses. The viewer spins the nose piece to select different objective lenses. So these three are different from each other because it has a different overlook. Okay, different objective lenses otherwise with this. Okay. Continuation. All right. Objective lens. So one of the most important part of a compound microscope, as they are the lenses closest to the specimen. So since we have this eyepiece, body tube, the arm, and the base, of course, we have this coarse focus and fine focus. This is the most important part. Although without the power, this will not work. This will not work. But technically speaking, all right, this is the most important part of the microscope because you are able to see with these lenses. Okay, you are able to see closer with the use of coarse and fine. All right. And slide stage, of course, this is where you place your specimen over here. Okay. And metal clips, adjusting clips, so you adjust on how to hold this specimen. And diaphragm, all right, adjust the amount of light that reaches the specimen. Okay, this is where you place your specimen using these two clips on the slide stage platform, and then underneath of this slide stage we have this diaphragm. So you can adjust from here, adjust the lights so that you can see this specimen. And the base is here, all right, connected to the arm and to the body tube. And also the light source, all right, through the power, it will open, it will turn on. The light source for a microscope, older microscope use mirrors to reflect light from external source up through the bottom of the stage. But now we have the latest update or the latest microscope with 3 objective lenses. However, most microscopes use low voltage bulb. All right, so it has only low voltage bulb with the use of the diaphragm to adjust the lights so that we can see this specimen through objective lenses. All right. All right, now this is your activity. Draw the microscope on your paper with the labels accordingly. Do some research about what if there is no available microscope nowadays. Are there some other ways that we can see cells? This is for you to find out. Do the drawing and writing on your paper. All right. All right. Thank you so much for tuning in with me. Hope to see you again on our next video lesson. Bye-bye.